All right, so I'm Hunter Strathman. Today I'm going to be talking about our development of a three-dimensional cell culture model for use with neural engineering applications. So before that, I just want to give a quick overview. I'm going to start off with a general discussion on just some very general neural engineering applications and the challenges with those applications with respect to their long-term use. And then I'll discuss how 3D cell culture systems might be an ideal in vitro model to try and develop new technologies to address these challenges. And then we'll go over an overview and characterization of a portion of the proposed model, and we'll wrap things up with some conclusions and a brief discussion on where we hope to take this work moving forward. All right, so implantable neural electrode arrays come in many different shapes and sizes. You have planar neural electrode arrays like you have over on the far left with these multiple shanks that have very small, densely spaced electrodes on their surface. It's sort of the classic microwire electrodes arrays like you see here with the TDT and then also the Utah electrode array. And so these devices are used for both recording and stimulating the nervous system. And they're used for a variety of applications, from trying to restore lost function to affected populations, to just basic neuroscience research to try and understand better how the nervous system functions. So one thing that's common to most of these devices and applications is there's some variability and inconsistency in performance throughout the duration of the implant. And so this is a study from a group at UT Dallas that looked at the long-term performance of the Utah electrode array implanted in rats. And as you can see over here, we're looking at the active electrode yield, so the percent of electrodes that are recording usable neural signals. We can see that's dropping off throughout the duration of the implant. And then looking at another metric for neural recording signal quality, signal to noise ratio, we see a similar trend here. And so there are multiple things that affect the neural recording signal quality. One of these is the innate immune response to these implanted devices, known as the foreign body response. And so one approach to try and alleviate this immune response is to decrease the size of those implanted devices we looked at before, and also, importantly, change the mechanical properties so that they're closer to the tissue in which they're being implanted. And the issue here is that there's really not a great in vitro setup to test out these prototype devices with these softer electrodes. So a lot of the times, these groups will move pretty rapidly into animal models where things just get much more complex and it can be difficult to troubleshoot a new prototype. And so really the field would benefit from a better in vitro model to more rapidly develop these new devices. And so we believe 3D cell culture systems provide such an ideal model for this application. And a 3D cell culture is essentially where the researchers have developed an artificial extracellular matrix for the cells cultured within them to grow and behave within a 3D environment that's more similar to their natural tissue environment. And it's been shown that cells cultured within these matrices behave more similarly to their in vivo counterparts. And so just looking at an example of this is a group that developed a collagen and hyaluronic acid-based hydrogel. You can see a cartoon representation of here, and they've encapsulated that with encapsulated Schwann cells within there, as you can see in this top picture. And then you can see a little bit better down here how those cells are interacting with the extracellular matrix from the scanning electron micrograph. And so what we're proposing to do is to use this technology to develop what we're calling a living phantom, specifically for neural engineering applications and to help prototype these new electrodes made of flexible materials. And so one of the first steps is to match the mechanical and chemical transport properties of the hydrogel to reported values for brain tissue. And then eventually we want to incorporate both a 2D and 3D cell culture system into the hydrogel. And you can see that a little bit in this cartoon here with the 2D monolayer on top, representing other tissue layers within the tissue, or possibly serving as a proxy for the scar tissue layer that builds up around electrodes during the foreign body response. And we also want to explore the potential to model some of these dynamic cellular changes that take place during the foreign body response by introducing various chemical cues or signals into the system. So here's a little bit of a zoomed in picture of that. And so here you can see we're making it about 2 millimeters so that researchers could actually implant one of their devices into it. and so that they can test whether the mechanical properties of their electrode hold up while they're implanting it into something that's very similar to brain tissue's mechanical properties. And not only that, because phantoms like that already exist, but they'll also be able to test whether it can actually record from neural signals as we incorporate neural cells into the gel. And so currently we're at the phase where we've developed the hydrogel and we are working to include cells into the model. So most of this talk will be encompassing characterizations of the hydrogel itself. All right, so now we'll go over a little bit of the formation process. And so to do this, I've modeled a cell plate insert that we can 3D print. That's based off commercially available transwell inserts. The benefit with this is that the experimenter can change these to whatever size or shape within reason that they want. And it's also substantially cheaper than those commercially available systems. And so to start off, we take one of these 3D printed inserts and we add a cellulose membrane to the bottom of it. And then we put it into a culture plate that's filled with calcium cross-linker solution. And then we fill that insert with liquid alginate and allow that calcium to diffuse up into the alginate for about 60 minutes. And this is the step that actually transitions from the liquid to the hydrogel. And then what you're left with is a hydrogel in this complete system that you can then switch out the liquid in there to your culture media or whatever you want to use for your experiment. And it's also worth noting that we've been able to make this hydrogel completely transparent. The hydrogel is actually in there in this insert. I've just removed the cellulose membrane there, which is going to be important when we start visualizing cells. All right, so one of the first things we wanted to look at were the mechanical properties of the hydrogel, since this is one of the key applications that we're proposing. And so to do this, I've looked at shear strain rheology. Over here, you can see the rheometer. This is the device used to evaluate these mechanical properties of the sample. And essentially what's going on here is there's two parallel plates that are rotated with respect to each other. And the lag between those two plates can be used to quantify or calculate mechanical properties of your sample. And so up here in yellow, you can see the shear modulus that we calculated for our alginate hydrogel. And then we're comparing that to some measurements that were done in rat and human brain tissue. And you can see we're pretty close to within the range of both of these samples are reported here. And these are some pretty preliminary characterizations, and we can modify this relatively easily, or it could be modified by any experimenter adopting this just by changing the calcium crosslinker exposure time. And so next we wanted to look at the electrochemical impedance. This is because electrochemical impedance spectroscopy is one of the most commonly used techniques for evaluating the electrical health of implanted electrodes. And it can also be used to track changes in the cellular environment immediately surrounding the electrode surface. And so we wanted to kind of co-opt this to use it to track monolayer formation, or as another method of tracking monolayer formation when we eventually incorporate cells. But before we did that, we had to have a good characterization of just the alginate hydrogel itself and to see if it changed over the time of a typical cell culture experiment that we would be doing. And so here you can see we're just comparing the hydrogel itself on the day it's made with just base culture media. And you can see there's very little difference between the two. And then, so if you look at it 7 days later, I don't know if you caught that, but there's a new line on there. And so there's very little change, which means it's pretty stable over those 7-day periods, which indicates that we could likely use this to track some changes in the cellular environment later on. And so next we wanted to look at the chemical transport properties of the gel. And this is important because we want to make sure that nutrients and waste will be able to diffuse in and out of the center of the hydrogel so that the cells within it can remain viable. And so to do this, we looked at the diffusion properties of a range of fluorescently labeled dextrins, as you can see here. And so the smaller dextrins had very good permeability, which indicated that the smaller nutrient and waste molecules shouldn't have any problem diffusing through the alginate. And the trends that we see with throughout the range of dextrins matches up with or similar to values that we've seen for reported in the literature for brain tissue. All right, so to wrap up, I've shown the development and the preliminary characterization of the alginate hydrogel that we'll use for the development of our full living phantom. We've covered the 3D printed system that's used in the formation and also just housing the gel for experiments. And then we've looked at the mechanical properties and the electrochemical impedance, as well as the chemical transport. And so next steps, obviously, are to start incorporating the cells, which we're currently working on right now. We want to, as I said before, we want to work in a cell monolayer on the surface of the gel, in addition to mixed cortical cells within the alginate, and hopefully be able to record from those neurons suspended within there. And then further down the line, we want to explore triggering these dynamic cellular responses by just coating the implanted electrode with rat blood or other chemical factors to try and recreate aspects of the foreign body response. And additionally, later on, it might be good to improve the sophistication of the hydrogel itself by trying to incorporate other cellular attachment sites or extracellular matrix components. And with that, I'd like to say thank you and thanks for your attention, and I'd be happy to take any questions. Yeah.
So my question is, how well do you think that something like this could scale to larger electrodes or tissue sizes? You talked a lot about microelectrodes, but what about like macroelectrodes?
Well, so for things like ECOG grid, I guess the size limit would be like surface area, like a wider gel, correct? Because their penetration wouldn't be that much different than some of the microelectrodes, right?
Yeah, I guess I'm just wondering how big of a like hydrogel do you think you could make so that you could measure things like that, surface grids that might be able to report deeper.
Yeah, that's a good question. For diameter, I mean, it's kind of limited by what you're able to rig up in the lab. I mean, cell culture plates, I mean, kind of seem to be limited around a few inches for the most part. You can get larger ones, but yeah, that would be. It tends to be the thickness where you kind of start to run into problems. So I would think that expanding in circumference or diameter would be relatively straightforward. But we've looked at mostly like around a centimeter in diameter. But yeah, that's a good question.
Thanks.
Yeah.
Great talk. I had a question about, so it seems you've spent some time really characterizing the mechanical properties of the gel without cells in it.
Yeah.
Do you expect that once you put cells in there, that there's going to be a significant change potentially in those mechanical properties? Is that like a concern that you might end up outside of that brain, region or that brain level of mechanical properties that you're currently seeing?
Yeah, I do anticipate some change for sure, especially as further on you get into the cell culture, if the cells begin to reorganize the hydrogel. At that point, we could try and do some just retuning of the mechanical properties, but yeah, that's kind of remains to be seen. It's a good question.